This method can detect the activity of retrotransposons called LINE-1s. These mobile genetic elements cause de novo insertions in cancer genomes and thereby contribute to genomic instability. The advantage of this PCR-based technique is that it is simple and cost-effective compared to high-throughput sequencing approaches. We demonstrate here its utility in detecting de novo insertions that originate from a LINE-1 inside the TTC28 gene on human chromosome 22.
LINE-1 activity can serve as biomarker for malignancy or pre-malignancy, especially in epithelial tumor types, like colon cancer or high-grade serous ovarian cancer. Besides LINE-1 insertions, this method can detect other genomic aberrations, such as DNA rearrangements that originate at rearrangement-prone region.
In order to select a suitable restriction enzyme and to design inverse primers, first download the TTC28 LINE-1 sequence in FASTA format from a LINE-1 database such as L1-base. The L1-base ID for TTC28 LINE-1 is 135. Include 5 kilobases sequence flanking both 5' and 3' end of the LINE-1 sequence. Export the sequence to a word processor. Here the LINE-1 flanking sequence is annotated in brown font and LINE-1 sequence is in gray font. To determine the unique tag, enter 1 kilobase sequence downstream of TTC28 LINE-1's 3' end into one of the polyadenylation signal prediction tools such as PolyADQ or Dragon PolyA Spotter.